Good evening and welcome to the second session of the Women in STEM Speaker Series, co-sponsored by the Research Initiative for Scientific Enhancement Program, better known as RISE. I want to thank my colleague Stephanie Bird, Assistant Director of the RISE program, for her collaboration on this event. My name is Tamara Pearson, and I am the Director of the Center of Excellence for Minority Women in STEM, charged with leading this groundbreaking initiative focused on equipping, empowering, and elevating women from historically underserved groups to thrive and lead in STEM fields. The Women in STEM Speaker Series is designed to showcase the research of prominent women of color in emerging STEM disciplines of interest to a broad STEM audience. And our session tonight is even more special because it features 4 distinguished Spelman STEM alumni who are actively engaged in making sure communities of color are not forgotten in this pandemic. I don't think we could have timed today's session any better. With the recent election of the first Black and Asian woman to the vice presidency, the spotlight has finally turned its focus on the astounding strength, intelligence, and fortitude of women of color. And if that wasn't enough, our Spelman sister, Stacey Abrams, class of 1995, was once again thrust into the spotlight as she helped to lead an organization that actively fights against voter suppression and whose work contributed to record voter turnout. But Stacey Abrams isn't an exception at Spelman, she's the rule. Spelman has a rich history of creating the future leaders of our nation, and the Center of Excellence for Minority Women in STEM builds on that strong foundation as we strengthen and deepen the knowledge and experiences afforded to our students and faculty. I don't want to take any more time away from this amazing panel, but there are a couple of housekeeping items. There is both a chat and Q&A function available for you to use. The chat can be used to dialogue with other participants in the webinar. However, please post any questions for the panel in the Q&A. We plan to leave about 15 minutes for audience questions after the panel discussion. And now we will begin our discussion with the introduction of our panel. To give you context for what each panelist brings to the conversation, we have asked them to prepare brief opening remarks. We will start with Jessica Coates.
Hello, everyone. I'm so excited to be here for this panel today. And when we were asked to come up with a few slides to kind of describe our journey in science and the work that we do right now, I wanted to follow in the theme that Dr. Corbett established 2 weeks ago when she really highlighted the role of mentorship and adequate funding for her to actually be able to go through the journey that she's been in and really highlight the role of African-American women, HBCU alumni, and HBCU faculty in my trajectory. I came to Spelman College because of an HBCU alum. I'm from a relatively small town where the neighboring HBCU is Fort Valley State, and a lot of the graduates of that university go on to teach in the area. And a particular teacher, her name was Dr. Veronica Sanders, she frequently told us about the opportunity to go to an HBCU, and at the time it sounded almost unbelievable, but you could go to a college surrounded by people that looked like you. There would be someone who would pay for you to do that, and they would pay you to do that. And so when I got the opportunity to be a part of the Women in STEM program, summer program, and academic year program that provided research support and a stipend at Spelman, I made sure to take an opportunity, as you can see in the upper left-hand corner. I made sure when I was at Spelman to continue to take advantage of all the opportunities that there were to participate in science and to do so funded as an MBRS RISE Fellow, where I did academic year research on antibiotic resistance and antibiotic proteins. And definitely took the time during the summers to engage in even more quantitative biology research at the Massachusetts Institute of Technology, the Howard Hughes Medical Institute, and even got the chance to go out to Snowbird, Utah, to really talk about the use of principal component analysis and how we can use that to find the differences between the immune response that's a result of our genetic makeup and our immune response that's a result of the environment that we're in. And so it really, it was really a no-brainer for me what the next steps would be. While I was at Spelman, I was a biology major and I minored in mathematics with an interest in infectious diseases. So I decided to go to graduate school at Emory University and focus in microbiology and molecular genetics. as a part of a biophysics team, I was able to actually understand what happens to bacteria when we subject them to antibiotics and develop mathematically validated mathematical models that could be experimentally validated for alternative treatment strategies for bacterial infections when antibiotic resistance was on the rise. And I also made the opportunity to step outside of the lab to really commit to global and domestic efforts for diversity, inclusion, and accessibility in science by being a part of an OpenCon Fellow program in the upper left-hand corner, and also visiting our nation's capital to talk with representatives about what we can do to make science more inclusive. In 2017, I joined the National Center for Immunization and Respiratory Diseases at the Center for Disease Control and Prevention here in Atlanta, Georgia. I originally joined the team as a contractor to help with outbreak responses. I joined in the middle of the Zika outbreak and continued to work with the team doing diagnostic and serology experiments for the Zika outbreak, measles outbreak, mumps outbreak, and I even got the chance to help with some of the studies in the Ebola outbreak, as you can see in this picture of me next to the Honeywell suit. But in the recent months, I have gone back to what kind of my original interests and loves are as far as quantitative biology and research, and taken on a predominantly research role on the team. And that role is really focused on trying to understand what transmission dynamics, or to estimate what transmission dynamics look like for respiratory diseases in different settings. And the way that I do that is by estimating this value that we call the effective reproduction number. And so this is the average number of secondary cases we expect to arise from an infected individual. And in my case, when there's a level of protection available, so it would be a vaccine. So if there's a vaccine with a certain level of effectiveness, what do the dynamics look like as far as disease spread, and how do those differ from setting to setting? I won't go into a lot of detail about this right now, just because in some of the upcoming questions, I'll give some more of the details about this work and how it's relevant to COVID-19. And so with that, I will turn things over to Dawn. I believe she's next.
Yes, thank you, Jessica. So next up will be Dr. Dawn Griffin.
Thank you, Dr. Pearson, for the opportunity to share a little bit about me and what I do. So let me just kind of start with how I ended up on this journey. Growing up in high school, my whole focus and thought was that I was going to go off to college and become an OBGYN. So I did make my way to Spelman College. It's the best decision I've ever made. And while at Spelman, I majored in biology, graduated in 1990, and took a gap year. Went on to Meharry Medical College and got my master's in public health specifically. And from Meharry, worked several years on several major projects there, including the I Have a Future program, which was one of President Bush's Points of Lights focus programs. And from Meharry, I went on to the University of Alabama at Birmingham on full fellowship to receive my PhD. So fast forward, once I was at UAB, I did work for a major pharmaceutical company, Aventis, now known as Sanofi Aventis, I believe, working in multicultural marketing. I couldn't believe that I was able to find some place where I fit in communication. And specifically, I worked on a project that dealt with type 2 diabetes, where we would go out into the community and sponsor different health fairs and, really kind of get a true understanding around what our community often refers to as sugar. So taking it from the sugar to the true diagnostic term of diabetes and how we could best manage that. And so that was really my first introduction into dealing with, outside of being a member of a community of color, working with communities in color and working to develop marketing messages that targeted Black and Brown people. And it was amazing. And, I'm dating myself, maybe that was 20 years ago. And it was amazing to me that we really had to be conscious, because for many in the Latino community, the decision makers in the household were women, but they didn't really speak English. So they were relying on their children to explain to them why product A, A, B, and C would work better for their family. So we wanted to make sure we develop messages that help communicate how products could be used for them. So once I graduated from UAB, I came on to the Centers for Disease Control and Prevention as first as a fellow, a health communication and marketing fellow through the Association for Preventative Teaching, and then as an ORISE fellow in the National Center for Health Marketing. Since that time, I've come on as a full-time employee at the Centers for Disease Control as a health communication specialist and scientist. Part of my responsibilities include taking everyday data and reporting those results to our various stakeholders through presentations, whether there be partner calls, different activities across federal agencies. For those of you that may not be aware, CDC is a part of Health and Human Services, and it's just one of the agencies that's under the umbrella of Health and Human Services. So we work across the different agencies. In addition to serving as the program lead for the National Conference on Health Communication, Marketing, and Media. I've had the opportunity to work on 2 emergency responses. Some of you that are on the call might not remember, but back maybe 11 years ago in 2009, we had our first H1N1 influenza A pandemic that we thought was going to really take everybody out. So I had the opportunity to work on that. Like Jessica, I had an opportunity to work a little bit on Zika. Zika hit at the time where our conference was going on, so I didn't get a chance to really focus in on that. And today, I work on the COVID-19 emergency response for my center. So at the Centers for Disease Control, I work in the director's office, more specifically in the office of the Associate Director for Communication. So let me say that anything that I share during today's presentations are my opinions only and not those of my agency. So from there, let me just kind of stop there and just say that there's some other things that I'm involved in that really kind of help make me, I feel, a better communicator, a better scientist. I am a member of the American Public Health Association. I'm a member of the Delta Honor Society, which is a public health honor society. I am a credential health education specialist. So I had that credential as a part of my training. I mentioned that I'm a graduate of Meharry Medical College. I also serve on the board of trustees for Meharry Medical College. And I'm also a lead mentor for the Sister to Sister program at Spelman. Specifically, I work with students in the medical, dental, public health, nursing, pharmacy group. So that's my plug for those that are on the phone who are interested in participating in Sister to Sister. It is a program where we match you up with different mentors that we find that are compatible with your particular interests. So what do I do on the emergency response? This go round, I work with a very small team who works across our entire agency. So I work on our liaison team for the Office of the Associate Director for Communication. I guess the gist of what we do, we are the traffic cops for all communication that goes out of the Office of the Associate Director for Communication. Oh, that's my boss. So we're responsible for making sure that as things happen so fast with this particular pandemic, that all materials that need to be pushed out to the public get cleared and reviewed before they hit the news waves, and we have to come back and pull something back. So often those of you that have been traveling, if you've gone in the airport, you've seen infographics with how to wear your mask properly. how to wash your hands properly. So, we design materials that are for the public to help them understand what's going on. In addition to that, we also review the data that comes in based on those that are in the field. We publish journal articles that talk about the different things that happen. One that I think some of you may remember may be the summer camp, the overnight summer camp incident that happened. So when that happened, we had to kind of get on it, come up with, what happened, the details of it, so that we could put it back out there so people would know and wouldn't have to really make judgments based on what they think happened. So in a nutshell, we are traffic cops. It's not hard science, but communication science is definitely necessary and needed, especially when you're talking about people from different walks of life. Communities of color. I used to use the example of when I worked at the pharmaceutical, industry. A term such as sugar is something that's very common, surprisingly, not just in African American, but in Native American communities as well. So, presenting information, given a scientific presentation that uses the official term or a scientific term, may not be so useful to someone who's accustomed to using local colloquialisms to determine what's going on with them. So it's important that we not only, produce materials that are at a certain reading level so that everyone can understand them, but also using some of the science, converting that into language that they can understand or frequently use language. So I think I rambled on enough there, Tamara.
No, that was perfect, Dr. Griffin. That was perfect. And we're going to talk more specifically about scientific communication a little bit later. So we'll definitely be coming back to that. So up next is Dr. Janina Jeff.
That's actually a perfect segue into a lot of things that I'm going to be talking about. So thank you so much, Dr. Griffin. I'm going to share my screen. Can you guys see my slides?
Yes.
Okay, sorry, I can't see you. Yes, so today I'm going to talk about, I want to start with talking about who I am. So my name is Dr. Janina Jeff. I graduated from Spelman in 2007 with a bachelor's in biology with a minor in Spanish. And right after attending Spelman, I should say how I became interested in science. So similar to, I believe this was you, Dr. Griffin, I started off my Spelman career wanting to be an OBGYN. And this was because my mother was pregnant with my sister my senior year of high school. So she was actually born my freshman year of college. And I foolishly thought I wanted to be an OBGYN. I say it's foolish because I do not like gross and grotesque things. And so I participated in a program called Mini Medical School at Morehouse School of Medicine, and saw my first cadaver and decided that that was not going to be the place that I was going to, that was not going to be the area in which I was going to impact medicine and science. But what I did do after that was join the RISE research program. And I joined this my sophomore year at Spelman and continued on into my senior year. And that was the first time I had lab experience. And it was the first time I was introduced into genetics. And so by training, I am a human geneticist. That means I study the genome, what I like to call the book of life, the DNA or the particles that make up who we are. And if you thought about your genome as a book, it's a book that contains all the recipes for all the bodily functions and things that you need to survive. And so I started my career in genetics right after I left Spelman and went on to Vanderbilt. I was actually the first Black person to graduate with a PhD from Vanderbilt. So I got a PhD in human genetics. And I began an academic career. So after I got my PhD, which was a 4.5-year process, I went on to do a postdoc at Mount Sinai and had a pretty heavy academic career. I have over 40 publications that are peer reviewed. About 20 or so of those are first author. And then in my third year of my postdoc, I realized that academia was not the right fit for me at that time. mostly because academia has its politics as well. And I know there are some academics on the call who can resonate, who this will resonate with. And so I learned really quickly in my postdoc that while I was interested in studying genetics, particularly through the lens of understanding health disparities and how our genomes make certain populations disproportionately at risk or not at risk for certain diseases, that type of research at that time was not being funded. And so I went on to choose a career in industry. So now I work at a company called Illumina, and we create genetic technology for two types of genetic technology, sequencing and genotyping. And so through this process, our company, we have customers like 23andMe and Ancestry. And actually, as a population geneticist, I design those tests by understanding population structure within the genome and making sure that we have representation in our products. And so my formal title is a senior bioinformatics scientist, but my informal title, and what I like to tell people, is that I'm a geneticist. And I say that because as a formal PhD trained geneticist, I understand the power of the genome and its infinite possibilities to yield the future of health, our futures. But as a cis, a Black woman, I understand the lack of ethical research and compassion for African descended communities. And really, my passion is how I can change this paradigm. And so one of the things that have became really apparent to me really early on in my career, that there was a gap in science communication. And so I would get emails from people all across the world saying, you know, we just need a woman bioinformatic person to come and speak to whatever audience, or we need a female geneticist or a Black female geneticist. And I started to have a career in public speaking. I actually accredit a lot of my public speaking. I'm not doing a great job now, but I actually accredit a lot of my public speaking skills to Spelman, because Spelman is a unique place where I feel like the science department, really a lot of us have soft skills, and people don't teach you how important soft skills are, but they become extremely important, particularly when you're trying to communicate science, but even when you are presenting science and doing science. And so as a result, pretty quickly, I became a science communicator. I didn't really start calling myself a science journalist until recently, but in 2018, this all started when I applied for a program with Spotify called the Sound of Bootcamp, which was geared for women of color who were interested in podcasting. Having never done a podcast a day in my life before this day, I applied, given all my grant experience, to write a 2-page proposal in 3 hours. Wasn't that difficult. And I was selected out of 18,000 applicants. I was one of 3 winners. And Spotify paid for my pilot for my first season of the podcast, which I'll talk about. But other platforms like TEDx Flatbush and Black cultural communication like The Root and the Karen Hunter Show have also been avenues in which I've been able to take what I've learned as a scientist and also give back to my community. And so I have this dual role of being a 9-to-5 formal scientist at a biotech company, but then in my off time, I do speaking engagements like this to communicate science to the community and make sure that we are all understanding and included in the science field, and particularly genetics. But I want to talk a little bit about the podcast. So I want to welcome you all to the family. I know my Spelman village is here and everyone has always been supporting me, but this is another village that I call the family, which is a part of the In Those Genes podcast. So In Those Genes is a podcast that uses genetics to decode the lost histories and futures of African descendants. We take Black culture and use it as a vessel to teach genetics education. And so we do this using Black culture analogies. Unfortunately, I won't have an example of that today since we're talking about COVID-19. But if you are interested in it, you can look for our podcast, In Those Genes, anywhere you listen to your podcast, as well as going to inthosegenes.com to listen for more. But since I've started the podcast, the first season came out earlier this year, we've been getting a lot of recognition. And this is really important because after attending Spotify, we became an independent podcast. That means we don't have any formal support in this, but we have been getting a lot of recognition from places like the New York Times and other science avenues like the American Society of Human Genetics. Our first season is all dedicated to genetic ancestry testing, mostly because we see genetic ancestry testing as being an entry point to the Black community, since genetic ancestry testing is heavily marketed towards us. But one thing I think is missing from a lot of what is available for genetics to the lay public is real communication and inclusion of not only just telling and including people in the research and getting people to participate in these tests, but making sure that they understand and making sure that they too become scientists and be able to understand and communicate science. So midway through our season this year, COVID-19 happened. And while we're a genetics podcast, and I can go on and on about the genome of SARS-CoV-2, I won't, but it was really important that we shift gears. So we quickly shift our podcast to start talking about COVID-19. And this happened midway through our season. So what I want to do is play a clip today that I think is somewhat relevant. This clip is coming from a recent episode that we had over the summer that talks about Black people and how we should think about taking vaccines. But ever since the mid part of our season, we dedicated a full episode to COVID-19, which actually just won an international award. That episode is called Dat Rona. But ever since then, we had 10 to 15 minute clips that cover different parts of coronavirus coverage. And all of our coverage is specifically talking about this through the angle of Black and Brown people. And like was said earlier, taking these complicated scientific terms and translating that to something that we can relate to, that we can understand, but that also engages us in a meaningful way. And so I'm going to play a quick clip and then I will end. And again, this clip is about COVID-19 and vaccines and talking about the possibility that vaccines could be made to target or hurt Black people.
Is it possible for scientists to create a vaccine engineered to specifically harm one group of people? In this case, Black people. This is similar to the myth we debunked about scientists creating coronavirus in the lab to do harm. In short, SARS-CoV-2 has over 30,000 letters in its genome, or book of genetic instructions. And what makes SARS-CoV-2 unique compared to the original SARS, the virus responsible for the 2002-2004 outbreak, varies only by a random shuffling of about 1,000 letters. The shuffle is so random that it's nearly impossible to orchestrate. Even if you wanted to create a deadly virus, let's say to hurt people, you probably want to go with something better than one where over 80% of cases just get flu symptoms and a loss of taste and smell. Ba-bum-bum. Psh. All right. Let's apply this same logic to the vaccine that will be engineered to kill us theory. When it comes to creating a bioweapon disguised as a vaccine, the genome will be equally important. Making a bioweapon that only targets one group of people who vary so much genetically is even more impossible and illogical. In order to develop a bioweapon that would specifically cause harm to Black folks and Black folks only, one would need to understand the interaction between coronavirus and the genomes of Black folk. And here's the thing. There is very little known about how coronavirus interacts with anyone's genome. Furthermore, because of actual systemic racism, very little is known about Black genomes in the first place. We can't even get medical treatments that work properly for us. So who in the hell got the research to create a bioweapon against us? We also know that, sadly, there are much easier ways of killing Black folks. It's our lived experience. The socioeconomic factors like poverty, increased stress, lack of access to healthy food and air are the bioweapons working against us. And they're not the same covert diabolic plan. They're right there for everyone to see and for us to experience.
So that's a clip from the podcast. That is probably one of the darkest clips from the podcast. I promise you, it's much more entertaining and light and fun. But I picked this clip because we're talking about COVID-19 today. And given the recent news around the vaccines, I think it's important that we understand how our genomes can be involved and could not be involved in the vaccine development. So this is my contact information. Feel free to reach out to me via email. I'm pretty available, and make sure you follow the podcast.
Thank you so much, Dr. Jeff. That's given me a lot to think about and a lot for us to discuss when we get into the discussion. But before we do that, we have one more panelist, Dr. Shelby Wilson.
Hello, everyone. It is a pleasure to be here. Thank you so much for the invitation. And I am going to talk to you for a few minutes about just my background in general and my relation to the COVID-19 pandemic. So, here we go. Of course. All righty. So again, Shelby Wilson. I attended Spelman College. I did not have a choice, unlike many of the women on this panel. I was told at birth that I was attending Spelman College, and I attended, and it was the best decision I ever made that was forced upon me. Very, very grateful for the forces that be that made that happen for me. I graduated from Spelman in 2006 with bachelor's in mathematics and computer science. I also, like many of the women on the panel, thought that I was going to Spelman to become a physician. And my mother sat me down my junior year and told me, Shelby, if you don't like labs, you will not make it through medical school. And so I had to come up with a backup plan. And my backup plan was to become a mathematician. After graduating from Spelman, I went to the University of Maryland, where I got a PhD in applied mathematics. I am what they call a mathematical biologist. There's all sorts of words, computational biologist, not quite a bioinformatist. I don't quite do statistics, but I studied, at the time, I studied how cancer grows and how we might better treat cancer. And that's where I kind of got my fill of biology, my fill of the medical world, also while fulfilling my love of mathematics, which was fostered at Spelman College. After graduating from the University of Maryland, I had the opportunity to go to France for a couple of years for a postdoc, and then I entered academia. I was on the faculty at Morehouse College for a number of years, and actually spent last academic year back at the University of Maryland in the Department of Biology. And I won't go too much into the fun of academia, but at some point I decided it was not for me, mostly spawned by COVID. And so where I am currently is I am currently a senior data scientist at the Johns Hopkins University Applied Physics Laboratory. And so this is a university affiliated research center, and affiliated with Johns Hopkins University. I have been there since May, so I have changed jobs during COVID. Honestly, when I found a place where my skills and my training could really foster and bloom, and as I tell my husband, I say I got drafted. So finally, after writing papers that 5 people read for many, many years, I got the opportunity to really have an impact on the world and what I saw around me and the pandemic, as related to populations that were in need and the need for people with my skill set to attack this current pandemic. Now, looking at what's here, I heard a lot of biology as majors, and quite frankly, during our virtual homecoming a couple of weeks ago, one of my very, very dear Spelman sisters came at me. She said, no disrespect, but what qualifies you to work on this, right? I am a mathematician. That is what I am at my core. And what I realized is that we being mathematicians do a very, very bad job at explaining what we do in real life and what are the things that we can do. And hopefully I'll be able to give you a little bit of feel for where these things might come together, specifically as it relates to COVID, but also in the array of many, many other areas of science. And so what I have been working on lately being the past 6 months, is actually modeling COVID-19 to inform humanitarian operations. So this is work that was done in collaboration with the United Nations OCHA office. So it's the Office for the Coordination of Humanitarian Aid and the Center for Humanitarian Data. And so this is what we've been doing. And the big idea here is that more or less, as this great worldwide effort in trying to understand what was happening with COVID and who knew where, this was one of the very, very few times where, quite frankly, the math and the modeling and the data were moving faster than the biology. Now we're starting to catch up, but at the beginning, all we had were numbers. And with those numbers, we had to figure out what to do with that. And so what we needed to know who was sick and where and when. We needed to know what our NPIs, our non-pharmaceutical interventions. So what can we tell people to do? How effective are they? If they're choosing one or the other, what to do? And we also needed to know country-specific features that affected the spread. So for instance, in countries where they don't shake hands, was the virus spreading less than in other countries? And so quite frankly, again, at the beginning, all we had were case counts, death counts, and somehow we had to understand and be able to project to answer questions, hard questions like, we have a plane on the tarmac with PPE, where should it go? That's a mathematical question. That is an operations research question that, quite frankly, at the beginning, we didn't know. These hospitals are filled, we have ventilators, where should they go? What should the messaging be? If we have this much money to put on this many campaigns, where should it go? And so these are the problems that I've been working on recently, specifically in the context of countries in receipt of humanitarian aid from the United Nations. And so the key factors in what I do, again, so kind of pulling together lots and lots of things here to try to put them into what we call a mathematical model. So most importantly are things like mobility. So again, we have a world that used to be very, very connected that now both locally, globally, regionally is now very much so disrupted. So trying to get estimates of who goes where and when, how often people are having contact with each other. So for instance, when you shut down a school, you no longer have school-aged children interacting with each other. How people are complying with the directives. What are factors that deal with vulnerability? So these are things in the case of COVID, like heart disease, diabetes, these types of things. Of course, exposure and data challenges. And the data at the beginning of this, now it's getting much better in certain places. We were reading infographics to try to figure out who was sick and where. All the hospitals were reporting data in different forms. You had no idea. They were counting different things. We had negative COVID cases all the time. And so just trying to understand what was there, who was reporting, and trying to get it organized in such a way such that we can actually make forecasts about what's going to happen 2 weeks from now. The idea is that we want to support operationally what is going on. And so what that looks like is that we have a model, and I won't go into the details of what that model looks like, but if you've taken an ordinary differential equations class, it's called the SEIR model. So these are models that tell how people get sick and when, just in groups. And the important parts here is that we have geographic networking. So we have connections by flights, by roads, things like that. How connected is one region to another, one county to the next. We also have to stratify our population. We know that COVID affects people of different age groups very differently. So we have to account for age differentials. We also have to account for health differentials. And in our case, we were very much so interested in food insecurity and indoor air pollution, because again, we're working in countries that were developing. And then just in general, we need epidemic modeling. So how many cases are severe? And once they're severe, do they have access to healthcare, right? And so those are the types of questions that we designed to answer with our model. And kind of what we came up with was a model that would more or less, again, tell you who was sick and when and for how long. When is the peak going to occur? How many deaths are we expecting? How many hospital beds are we expecting to be needed? And how many people were not going to have access to those beds? For instance, some of the countries that we modeled in, there were 10 ventilators in the country. Well, once those 10 were taken up, no one else got any. And then more specifically, our questions were, what could be done, right? So given a limited resource setting, what are the things that we could put into this model to see how they affect the spread of COVID in these countries? Those were things like modifying social modifications. So again, these are things like restricting sizes of gatherings and things like that. Access to healthcare. Can we encourage people either to go to receive healthcare or not go to receive healthcare? Can we set up temporary hospitals? There are things like limiting geographic movement. So this is a map of South Sudan here. So for instance, can we cut off one region and prevent mobility in and outside of that region to try to contain the spread of the disease within a specific location in the country? And also the idea of medical and humanitarian intervention. This is ideas moving people from the vulnerable class to not vulnerable. And that can either be by treating them, their other medical conditions or other surroundings, things like giving them access to hand washing stations. So moving them from a position where they're more vulnerable to the virus to where they're less vulnerable, susceptible to severe cases of the virus. So essentially we throw all this into a bag, shake it up, and come up with this model that gives us projections. And so what that looks like is that looks like reports that go to the WHO country offices in the countries. We give projections of how many cases we expect in the next couple of weeks. So we project about 4 to 8 weeks out. How many deaths do we expect? And more specifically, where do we expect them? So this case is a case of Afghanistan, one of the countries that we modeled in. And so again, we have reported new cases, and you can kind of see the spread of where the disease will be and when. Also, for instance, this province right here, this is the province that lines with Iran. And so kind of when all of this happens, there were a lot of displaced people who were in Iran who came back to Afghanistan. So they were coming back to their home country. And so in this area, which also is included in the model, there's a lot of internally displaced people. And therefore that's kind of what leads to the higher daily rates and the higher death rates, because we have a particularly vulnerable population in this region here. So those are the types of insights that we get out of the model and what we have. This model also is included in the CDC ensemble. So we do projections here in the United States. So we are a part of that group too, where we model basically every county in the United States and exactly the same context, and make projections there. There we are part of a larger, as I mentioned, ensemble. There are a lot of academics who create these things, and kind of together we try to make projections about the current situation of COVID and what's coming up in the future. And that's what I do. So in case you want to know, this model is called OCHA Bucky. OCHA for the Office of Coordination of Humanitarian Aid. Bucky is the name of the dog of one of my colleagues. So we had to name it something where it wasn't associated with any one of us. So it is named after his Cocker Spaniel. But all of this code is now publicly available. We were very excited. We had a big event with the United Nations a couple of weeks ago where we got to do a kind of a public release. All the reports are there. So right now we are modeling, as you see over here, in Afghanistan, DRC, Iraq, Somalia, South Sudan, and Sudan. We have the capabilities, quite frankly, if we get good data and you give us a country, we can make it happen. And in particular for me, this was nice because amongst the big push to, number one, get modeling upstanding at the beginning when we didn't know what was going on. But this was the opportunity to work for a population that is often overlooked and often vulnerable. All of the first models you saw were out of France, Italy, Spain, UK, US. And quite frankly, a lot of people were lost in that big rush. And so in taking the time at the beginning to focus on these vulnerable populations, now I feel very good about that work. And now that I don't want to say that we're having the opportunity to breathe now, but now that we have a better handle on what we need and when, we are able to start to incorporate things in the US, such as demographic information and things like that. And with that, I'll say, feel free to shoot me an email anytime, shelby.wilson@jhuapl.edu, and I will turn it back over to you. Thank you.
Great, thank you. So as you can see, we have an amazing panel of Spelman alumni. So we're going to jump right into our discussion. So for this first question, I'm actually going to start with you, Dr. Jeff, and then everybody else can jump in after. So the first question is, when thinking about the COVID-19 vaccine development and dissemination, what should communities of color know and understand?
Yeah, I think that's a good question. So one of the things that I think there's a lot of fear built up, and I think that fear is justified, given the way and the history in which we engage with the medical community. I was on a talk a couple of weeks ago, and we talked about where does that history and that distrust start from, and what has been the history of our engagement with the medical community. And Black Americans, our history, when we first came to this country, we either saw a physician because we were sick and needed to go back to work. So we were so sick that we couldn't work, or we were used for research for the benefit of other people. And so that is the history of how we began to engage with medicine. And of course, that has changed over time. But that mindset has not changed as much as it should and could. And a lot of that is based on the lack of trust that has formed because of those two ways in which we engage with the medical community. One of the things that I talked about in my introduction was the fear that a vaccine could be created to intentionally target one group of people. That is a myth that just simply isn't true, and from a genetic standpoint is nearly impossible. One thing that people should know, and there are a lot of resources, but our podcast is one, but there are a lot of resources that talk about the vaccines and what type of vaccine. So I think our community needs to, one, know the possibility of a vaccine being created as a bioweapon is very low, if at all. And then also, what are the types of vaccines that are out there, and looking at the data to see how all of these clinical trials and all of this data is public, how they are doing, how diverse are the samples that they are testing these vaccines in, and what type of vaccine would be the type of vaccine that they might want. And so a lot of people ask me all the time, well, can the virus enter your genome? There are different types of vaccine, and the traditional flu vaccine is a good example of a live attenuated vaccine. That means we expose you to the virus at a dose that your body recognizes it and creates the antibodies that you need in order for you to not get infected and get the disease. But the two major vaccines that are being developed so far are not live attenuated viruses. They do not enter your genome or do any manipulation to the host genome, your own workhorse that you got going on in there. And so I think it's important that we understand the types of vaccines that are being offered, the risks that are associated with those vaccines. This is all public information and things that avenues like the podcast talk about, and access to the vaccine. So I encourage everyone to not take a vaccine really until you understand what a vaccine is doing and what type of vaccine is best for you, and also thinking about which one of those vaccines are accessible to you. But in general, excuse me, in general, I think another thing is to understand is what are some of the criteria in which you should evaluate which vaccine you want to take. And so I talked about the different types of vaccines. One's like a live attenuated vaccine, which is exposing you to the virus similar to the flu. One of the reasons that, and just a tangent here, one of the reasons why the flu vaccine is half as effective as when you get it is because the flu is a virus that is consistently mutating, and that mutation will be big enough that you will still get a different manifestation of the flu. That is not the case for COVID-19 from a genetic standpoint. While the virus is mutating, it's not mutating in a way that you're getting a completely different, type of exposure to a different virus. It's not that different. One of the ways that we've been using the mutation is to kind of track where the virus is going and see how the virus is moving across the world. But it's not really changing the pathology of developing COVID-19. What I was going to say, so yes, learning about the different types of vaccines, learning what vaccines are available to you and where you can get access to those vaccines. And you can see who is supporting those vaccines, who is funding the research behind those vaccines. All of this is publicly available information.
Great, thank you. Dr. Griffin, did you want to jump in?
Yeah, if I can piggyback on what Dr. Jeff shared, I think she hits it right on the nail. I think that we have to encourage our parents, our grandparents, our cousins, all of them to be their own consumer advocate in this situation. We're in Black Friday season, and I have seen people really put in the research to determine what type of television they need to purchase, whether or not they can see, how many pixels are on it, how many, I'm not a technical person, but, all the bells and whistles on that. And I think that in this situation, we're going to have to educate our communities to do the same type of research when it comes to determining which vaccination is going to be the best for them. And understanding the risk and how will you have access to it. I listened to an interview earlier today that was talking about one of the vaccines that's coming out, and they were just, asking a young lady, how did she feel? She's in the Pfizer trial right now, and it's a 2-dose trial. So, there's going to have to be some education in there so that that if you're doing a 2-dose trial, you don't get to take just the 1 dose and think that you're going to be okay. You're going to have to be committed to following through with whatever that regimen is going to be. So, I encourage us to make sure that we encourage our families to be their own consumer advocate here. We research electronics, we research, what type of shoes we want to go purchase, what type of car we want to buy. We have to make ourselves a priority, especially when it comes to this. And I think that we are also going to have to encourage our leadership to be transparent in this so that we know what's really happening, what the side effects are going to be, how will we have access to it. So yeah, I think we have to hold everyone accountable. And I think that as there is a shift in the days to come, there's going to be a lot of information that's going to be put out there. So we're going to have to also encourage to ask the questions if they're not familiar. You know, a lot of our more senior family members and colleagues will just go on a recommendation because such and such said it was good. I'm going to go and get it. So we're really going to have to get them to really kind of be in trying to determine what's going to be the best fit for them, putting it in plain language so that they understand when they go to the doctor, this is what I'm committing to.
That's a great segue into the next question. And I'm going to start with you, Dr. Wilson. What are the responsibilities of the scientific community to translate findings for a general audience so we can make those informed decisions?
It is incredibly paramount to for the scientific community to speak in a language and to display information in things in ways that are transparent and that are understandable. And I think that, again, going back to what makes you qualified to do this is that quite a bit of what we do as scientists is learning how to communicate with each other. And what is rare is learning how to communicate with the public. And there's always the question about how much information should be shared with the public and who knows what and when and things like that. And I think that it is absolutely paramount that we learn how to communicate that science is not voodoo, right? Like that there is good thought and quite frankly, give the limitations of it as well. And I think that part of the problem right now is that there's these kind of absolute statements that are coming out. And in reality, what is happening is that the public is watching the scientific process in real time, right? And that often doesn't happen. Usually we sit in our corners and we come out when we're ready to say something. And right now what you're seeing is an iterative process. For instance, I saw an article the other day that said, you know, the government's been hiding hospital data for, you know, months. And I'm like, no, we just didn't have it. Like it wasn't being collected. It didn't they weren't collecting how many Black people were having COVID at the beginning. So we can't say things because it just wasn't there. So I think that as we move in, especially in this next phase going into this third wave, which we're clearly well into at this point, and for the hope of a vaccine, I think that science communication is incredibly, incredibly important. And it is on the burden of the scientists to make sure that we display our information in ways that is consumable by not only the public, but by the decision makers. Often our decision makers are policy makers who are not scientists, and to hold that part of our responsibility very, very near and dear to our hearts, and to take that responsibility very seriously.
Great. Jessica, do you have anything that you would like to add to that?
Yes, I completely agree with Dr. Wilson. As a part of the work that I do, I try to identify where susceptible populations are as a consequence of whether or not they've decided to either be compliant or accept whatever the coverage method is. And most of the times when there's a mention of not accepting the coverage method or being reluctant to accept that coverage method, it has to do with how much trust there is in the scientific community and what they're doing. And so as a part of that OpenCon fellowship that I mentioned, one of the big initiatives that we take on is this idea of openness in science, and that science should be open on multiple levels, especially science that's publicly funded and driven by the dollars of the people. And so one of the things that we believe in is that when scientific research is published, it should be published without a paywall. So that should be open access. When we're developing these models and making conclusions based on data, that raw data should be included and should be publicly available for not only the scientific community to critique it, but also for the individuals who are going to be participating in these different preventative methods, for them to be able to look at it and understand what's going on. And then lastly, I'm also a huge proponent for open education resources. So these are resources like free online courses, textbooks that are cost effective, and so forth, so that if the public decides they want to get information and they want to learn, this is epidemiology, public health, vaccinology, biology, mathematics, all these different areas that are coming together to provide information are really wide. And even myself, I know this much about this much. So having the opportunity to get education and get knowledge without financial access being a big preventative is something that I think we should really push for in general. And then when it comes to the Black community, as Dr. Jeff mentioned, there have been multiple egregious acts in biomedicine and in medical sciences in general with Black communities. So outside of that transparency, we need to take that additional step to engage with the community, engage in conversations like this, talk to our family members, talk to our friends, provide any and everyone we can with as much information so that they're well equipped to participate and understand these conversations, as well as share that information. Because when it comes to public health, if you don't have public buy-in, we're not going to see transmission really decrease, and SARS-CoV-2 and COVID-19 is going to be something we deal with for a long time.
If I can piggyback on what Jessica said, I was really thinking about, how do you increase vaccine acceptance in the community of color? And I think it's going to mean for us having key stakeholders at the table, serving on those teams, not just hearing what Shelby's doing at Johns Hopkins. It just got me really excited because, all I've been hearing, for the past 9 months, well, Johns Hopkins is saying this. And so I feel confident, with the science that's being, judged here. But I think we're going to have those key stakeholders, community leaders that are at the table, whether it's arguing for inclusion or arguing for the transparency. We're going to need to have the physicians, groups like the National Medical Association, the nation's, oldest organization for Black physicians, really involved in this so that they can help put those messages out, develop the messages that need to go out and help push them out to our various communities. We see, we saw what happened with the elections and how we were able to get so many celebrities out to really talk about the hardcore. We're going to have to try to determine how can we pull some of those famous faces into this equation to help explain what needs to happen for us to, to be ahead in this whole scenario. I just had a thought. I was thinking about Alex Trebek and from Jeopardy and how I don't know if it's the Pancreatic Cancer Association or one of those associations has just in the 2 days that he's been gone, they have seen a tremendous increase in dollars for funding for curing pancreatic cancer. We need to have, that type of rock star push on this in order to get peoples of color to pay attention to what's going on. Oftentimes we pay attention to people when they look like us or they come from our experience. And so it's going to really take, we need the scientists in the labs doing what they need to do. We need what did Shelby call herself? The mathematical biologist doing, what she's doing, but we're going to need some everyday people that are going to be willing to take this on and making sure that they're spreading the word on what needs to happen in this.
That's such an important point. And Dr. Corbett, when she spoke a couple of weeks ago, she talked about that part of why she, works tirelessly to speak in so many different places is that she knows the impact of, Black people seeing a Black woman leading up these efforts and how that influences their decision to trust, what comes out of this vaccine trial. So, Dr. Griffin,
I'll be honest.
Yeah.
One of the scientists, I believe, on President-elect Biden's COVID-19 responses, she's a woman of color. I think she's from the Dominican, maybe. But I got excited when I saw her, because I was like, okay, we got a seat at the table. So we got somebody who's going to raise the questions and ask the questions that we can't necessarily ask. We'll let them know that Dr. Jeff is available if they need her, on the team. But
I'm sorry, I had a UPS delivery randomly come. Did you ask a question to me? Sorry.
No, I was just saying that I was excited about the transition teams, COVID-19 team that they have stood up, and that there is a young lady that's on there who's of Dominican Republic, and she's one of the scientists that's going to be working as they stand up their team for next year. And I was just volunteering you to serve on the team. That's all.
And Vaughn, I think you touched on something that's really, really important as well. So also there's like the community, like us getting vaccinated, us protecting ourselves, but there's also us having a seat at the table where the decisions are being made. And I know that I sat in rooms silently for weeks, and finally I just couldn't tell. I was like, we're not getting that vaccine. And they're like, what? I was like, we're not waiting for no vaccine. Like, I don't know a single person who has told me that they are waiting on. And they're like, and I promise you, I sat there in, daily meetings all day, every day. And it was 2-3 weeks. And I was the first person that anybody in that room had ever heard say, oh, nobody, that's not our experience. That's not what my Twitter feed is saying, right? And really, then the focus could change. Right? I wouldn't say change completely, but also what it does is it informs, again, decision makers that, okay, just making a vaccine available is not all that needs to happen, right? And they don't know that unless some of us, my Spelman sisters, anyone else comes and tells them that this is not our experience. And in particular, this came, again, like I said, I've done most of my modeling in developing countries, where you talk to the Democratic Republic of Congo, and you're talking about this vaccine. And meanwhile, in France, you have commentators saying, oh, we should test the vaccine in Africa, right? And when that's perfectly normal and perfectly acceptable, and then you talk to people on the ground and say like, oh yeah, you should get a vaccine, they're saying no. And I think that that is the importance of having diversity in the rooms where decisions are being made, because not all experiences are the same. And quite frankly, people don't know, they don't know. And I'm hoping for goodwill for the people that once they do know, that they can then take the action, because to encourage us to do what we need to do in order to protect ourselves. But that doesn't happen if they don't hear our message, if we're boxed out of the rooms. So.
Yeah, the whole idea regarding distrust is something that people, for some reason, think that has just gone into the back of our memory, and it still doesn't exist. The Tuskegee study comes up to this day, and it will live on forever. I mean, we're 70 years, 80 years, since that happened, and I think that we're all, in the back of our mind have that thought that Tuskegee could happen again.
Dr. Jeff, did you want to jump in?
I just wanted to say something, and it's kind of detailed, but I think it's extremely important, is understanding. So after you do all your research on the vaccines, try to do some research on understanding the approval process, because during COVID-19, we have seen a lot of FDA clearance, and what you've seen is this FDA EUA clearance, which is the emergency use clearance. And we saw it fail earlier this year when hydroxychloroquine got FDA EUA approval, and the first vaccine is estimated to get its FDA EUA approval later in November. And so when people hear FDA approval, I just want to reemphasize, it's important that we understand exactly what that is. And that goes to doing the research. Another thing, I'm going to drop a link in the chat, which is the link in which you can track all of the vaccine trials as well as the COVID-19 treatment trials. And in this link, you can have access to the different data that is all publicly available. And you can see all of the things that I would say are important to know about different vaccines, but also treatments in case you do become infected with the virus.
Great, thank you so much. So before we get to the audience questions, I have one more question for everybody, and I'm going to start with you, Jessica. So as a Black scientist, can you talk about being the only or the first, as well as any added responsibility you feel to look out for communities of color?
Yes. So thankfully, for most situations, I haven't been the first. And a lot of rooms that I enter into, my Spelman sisters have already kind of established what the tone is going to be. Sometimes I'm happy about that, sometimes I'm not, because we have a lot of really high achievers coming out of Spelman College, but there are very frequent times that I'm the only. And for example, in those pictures that I kind of showed in my graduate school research and some of those efforts, I definitely was the only. Having an interest in quantitative biology and especially in physics, this is definitely a male predominated field. I probably, if you gave me 24 hours, I could probably name all the Black women in this country with a PhD in physics, and I might know a large percentage of them as well. And so entering to rooms, I'm oftentimes the only Black person. And then something that comes out quite frequently when we're doing data modeling and statistical analysis is that missing data is something I deal with frequently and commonly. And so when data is missing, sometimes it can be missing just at random. The studies that I look at are largely observational studies. So once the vaccine's in place, we wait and we see what happens as far as who gets infected and who doesn't get infected with the virus. And so when that happens, there are certain biases that occur. And when people are trying to collect that data, there are limitations to doing that. So there will be missing data. But at other instances, the data that's missing, it's not necessarily random, but it's associated with why this particular population may not have wanted to participate in the study. That could be associated with the study design, who was implementing it, how it was implemented, was it accessible to the population that we're interested in? If it was accessible, was the experience something that made them want to continue in the study so that we could get that longitudinal data that we need to make the conclusions that we're hoping to do? And oftentimes, there are quantitative methods for addressing missing data. You can use methods like looking at the mean or the median for the population and assuming that where the data is missing, they look just like the rest of the population. But as an African-American woman, I can say for certain, my demographic and the data that comes from me may look extraordinarily different. And that's something that is a motivation for me. And in the work that I do is to not only, if there's missing data, to take that extra step to figure out why the data is missing. And that extra step involves reaching out to investigators who are involved in the studies and asking them what's going on, getting that qualitative story and that information about why it's missing. Because once we understand why that data is missing, we can better inform those outcome decisions. And something that, I don't know, as a Black woman, and why is this so important to me, I think about it oftentimes when it comes to maternal health in this country, right? As a Black woman, the risk of dying while giving birth is much higher compared to other women in this country. And there are data studies that you can look at an outcome and say, well, part of this reason is associated with the fact that Black women don't report pain often. And one of the reasons why people can make assumptions about why Black women don't feel pain or don't report pain might be because we're superhuman, right? And this dates back, as like Dr. Jeff mentioned, to the start of OBGYN, right? A lot of times in certain settings, there are individuals who may not want to speak up. They don't feel comfortable speaking up. They don't feel comfortable with the individuals they're dealing with, and they don't feel comfortable in working with the study. So their story can become missing overall. And so as a Black woman in science, and frequently as the only Black woman, something that's super, super important to me is making sure when there's data missing, and especially if it's data that might impact the community I identify with, that I take that extra step to really make sure whatever strategy I'm using for replacing that data is informed by the investigators and the community that I'm most interested in.
So I'll speak to being one of, I won't say the only, I'm very grateful for the fact that Spelman College has given me the blessing of not being the only for in very many settings. But something that Jessica said sparked. So the year that I got my degree, that I got my PhD, the AMS, that's the American Math Society, publishes who gets PhDs and all of their demographics. And so out of 3,000 PhDs, there were 9 African-American women. And I saw that number 9 and I immediately named the other 8, right? They got PhDs the year that I got them, right? It should have been a star because I could name them all personally, and probably for the year ahead and year behind as well. And it's hard, right? Like it's hard to be in spaces where you are the only one. I have shown up in the publication for SciAm for conferences. Somehow my picture always ends up in there because they certainly take my picture every conference I am, because I'm the one little dot in the room, me and a few others. But what I will say is that I am grateful for Spelman College. I'm grateful. I did not know I was doing anything weird when I was there. We had culturally relevant science, and it was never a thing that I was a woman, that I was Black, or any of that. And it is amazing how much you can learn when the fact that you're a woman and the fact that you're Black are foregone conclusions. And what I cherished was the time when math was kind of all that I had to worry about, and I had people around me who felt the same way. And I would say that I have been very, very encouraged to be able to find those communities everywhere that I've been. I've had to seek them out. They are harder to find, but my experience at Spelman has taught me that I need that. And that has come from fellow Spelmanites that I've graduated with communities that I've been able to build since then. I will put a plug here for my website. I have a website called Mathematically Gifted and Black. Me and 3 other friends, every day in the month of February, we feature a Black mathematician because we do exist, right? Like, and that's the whole point. And building these communities and being able to walk in these spaces and to know that you aren't the only one and that there are people who are behind you. I think about, of course, Stacey Abrams. I think about Rosalind Brewer. I think about the machine that is behind us, and I'm grateful for that. And it allows you to be the one and only without feeling lonely, and I'm grateful for that.
Thank you, Shelby. Dr. Griffin and Dr. Jeff, do you want to respond to this question?
Sure, I can. I was the first and only at Vanderbilt, which was unusual. I will say my program is fairly new, and I do think that when I graduated from Spelman, it was around the time where computational biology and computational sciences was up and coming. I don't even think data scientist was a formal job at that time, but I have been one of many and few. I would say one of the biggest things that I have realized is that I think a lot of times there's this dual feeling of being proud and also feeling sad. Being proud to now have representation, but then also feeling sad. And I went to a talk at a high school, and a young woman was writing a book about Black women in STEM, a high school student, overachiever. But she had mentioned a term, and I can't remember the term, but the term was around how when you are the only one and there are not a lot of people like you, how you now take on the responsibility of all of that demographic. And you carry that weight of that demographic. And I think that is so unnecessary for us to try to do, and probably impossible, and probably also wearing us down. And so one of the things that I would say is useful is 2 things. So one, in that regard of like carrying all this weight, remember that this is not your weight to carry, that the reason why you are the only one and the reason why you are the first one is not anything that has to do with you, nor are you a monolith or a representation of all Black people, of all Black women, of all Black scientists, etc. So do not carry that weight. And another thing though, is being the only one or one of few and people who come after me, it's really important that we share our stories. My story wasn't, a straight A story. There were a lot of bumps along the road. There were a lot of things that got in my way. There were a lot of things that I thought that I didn't say, but I didn't have the words for, like imposter syndrome. I didn't know what that was, but I was dealing with it for a long time. And I think it's really important that we tell our story, not just the nice things and, I accomplished this and I achieved that, but we also tell the other parts, the days that were dark. Those are the things that the women who are coming behind us are going to be able to relate to, and they're going to be able to better deal with them as we share our stories and our experience. Because at the end of the day, one of the bigger things that delineate me from my white counterparts is that my story is very unique. My story is very layered. My story is this intersection of being Black, being woman, and being a scientist, and all these other things, right? And with that, it's not always easy, and it comes with a bunch of complicated things. I talked about being a geneticist, well, it really kind of stemmed from me feeling like I had this double consciousness, like W.E.B. Du Bois talked about, of having two Black experiences and having to navigate all of that. Sharing those things are really what resonate with the generations to come. And so that when they face it, they're like, oh, this is not new. Someone else has already done this. Janina did this. Jessica did this. Maybe I can reach out to them and talk to them about their experiences. And we can talk about the things that we wish we would have done differently. Because there's a lot of learning in that too.
And, I'll share a couple of thoughts, Dr. Pearson. I attended an Irish Catholic high school, and I could count the number of brown faces on one hand. And so I was determined to come to Spelman College. I only applied to one school. Granted, I really wanted to. My cousin attended Morehouse, and that's where I thought I was going. And they finally got me to understand that Morehouse was an all-male school. So I applied to Spelman and was accepted. And Spelman pulled out in me the confidence to walk into any room as the only woman or the only, African-American woman and not, and be able to hold my ground. So I can say outside of my experience in pharma, I've been very confident going into the room, not because I felt like I had anything to prove, just that it was in my DNA that I could handle any situation that was put in front of me, regardless of what my skin color was or the skin color of those that were in the room. So I haven't encountered too many opportunities where I've been the only one. Public health is rich with a lot of Black and brown faces that are the boots on the ground working. We are in public health, but I think that for me, the bigger thing for me is that if there are opportunities that I see out there, I feel it is my responsibility to recommend someone with a black or brown face to serve in the capacity. So one of the projects that I work on is a major conference. And I would often hear, your conference is very, white men, older men. So I'm very intentional about searching for those that can be at the table that can share, more about their experience, because they are the boots on the ground. So I think that's my role and responsibility as a scientist is if it's not me, who can I recommend to have that seat at the table?
I love that. Thank you. So we have a few questions from the audience that get back to the science of COVID. So the first question says, is it safe to say the virus has mutated at this point? It seems that people are not getting as sick as compared to the beginning of the pandemic, i.e. just quarantining at home and seemingly bouncing back. Yes, cases continue, but the mortality rate and or need for ventilators seems to be flatter when compared to the early pandemic. So who would like to tackle that?
I can talk about it and pass it on to someone else. So I saw this question. I want to just reiterate from a genetics perspective, and my company is doing a lot of the sequencing and providing the technology for this. The virus is mutating, but at a much slower rate than something like the flu. And the mutations that have occurred are not in any way connected to the pathology of the virus. I think what we're seeing though, is a testament to all the amazing research that has happened in the last 6 months, and how now we are becoming, and definitely not there yet, somewhat of a well-oiled machine when it comes to treating COVID patients. And there are plenty, I don't think there is a healthcare network or hospital that I know of that doesn't have a well-implemented plan of action when patients come into the clinic. And I would say that even goes for people at home. And the doctors who are seeing patients who are self-treating at home. I had the virus in April. I self-treated at home. And, I did have antibodies. I no longer have antibodies. So that doesn't mean that I can, go out and just be around a whole bunch of people and never contract the virus again. That is rare to contract the virus again, but it has happened. And so I think what we're seeing is not really that the virus is mutating or a different form of the virus, but what we're really seeing is how well we've become at treating the virus. And the drugs that have been FDA approved and are giving to patients, and things like convalescent plasma and other methods of getting people antibodies that we've seen over the coming months.
So I'll speak briefly on the logistics. So I'm, again, not a biologist. So the mutation, I hear from people like Dr. Jeff, but I can say that it does seem that we are getting better at treating it. It's interesting because something that was perceived as heartless back in May and June, we were trying to tell people to stop putting people on ventilators. The number one indicator of death was being put on a ventilator. And so essentially stop spending money on ventilators, spend money in other places, because it actually, that is not a good way to treat people to get them back to health, right? And unfortunate, but it is a lot of resources and to maintain someone in that condition and to get them back to health. So I think that kind of putting our resources in the places where they are most useful has been helpful. I think that people have been better about themselves recognizing the symptoms and kind of being able to start treating earlier, which is helpful, and aren't waiting until the last minute to show up at a healthcare facility. Whereas at the beginning, I think people were really trying to stick it out at home, quite frankly, too far, too long. And it is true, again, from the mathematical statistical sense that the death rate is quite a bit lower now than it was back during the first and second waves, which is encouraging. However, at the rates that we're getting diagnosed now, it doesn't help much if you have twice as many diagnoses. We are quickly approaching any advantages that we've gained in treating it by overcoming that with new cases at this point.
Great. So the next question, and I think this question is about the science, but also hits a bit about how we're communicating information. And Dr. Corbett talked a bit about some misconceptions that people have about antibodies. So the question is, if someone has already contracted COVID, would the vaccine be necessary or beneficial to them?
Are you gonna go, Jessica?
Dr. Jeff, you go first and I'll follow up. How about that?
So yes, I can just say that from experience. So I just told you guys I had COVID. I had antibodies up until September. And in September, I no longer had detectable antibodies, I should say. And so people who have had COVID should definitely still get the vaccine. There have been some cases of reinfection, and reinfection is actually confirmed by seeing 2 different strands of the virus. So the mutations that we were talking about. So it is definitely important. You still need the vaccine, because even people who have developed the antibodies may not have developed enough antibodies or the right antibodies to fight off the virus if they were to become reinfected again. So it's still important and beneficial to them. And in fact, in the clinical trials, they are running these tests too. They are running tests and giving vaccines to people who have already had the virus and people who are being re-exposed to the virus. That's a very important question. I'm sure Jessica has more to add.
Actually, no, you did a wonderful job, because I was going to mention, when it comes to infections with viruses, the immune response, it does vary from person to person. So just because you have a natural infection doesn't mean you necessarily develop antibodies that are able to neutralize against future infections. And then also over time, the immune response begins to wane. And so if you previously had SARS-CoV-2, right now, at least from what I know, it might be a little bit difficult to detect if you actually are protected. And chances are you may not. So to be able to protect yourself and those around you, I would definitely suggest getting the vaccine again, or getting the vaccine once we understand the safety, the longevity, and so forth.
So just to kind of ask another question related to that, because what I hear is, this is a common misconception that is out there that, I hear it in my family, like, oh, I think I already had COVID, so I don't need to worry about this. So, Dr. Griffin, as a scientific communicator, how do we shift that thinking?
When you said that, my first response was, maybe not the best response, was like, this is not chicken pox. It's not like, you've got it, you've had it once, you're not going to get it again, because, a lot of people thought that. And then shingles came along. If you've had chicken pox, you're more prone to get shingles. So I think it's going to be just putting it out there. We have a whole lot of myths for a lot of things. Taking vinegar every day is going to cure your sugar. I think we just have to put it out there and educate and put those messages out there. Make sure, the stakeholders are, repeating those messages so people understand. You may have those antibodies, for a few months, but it's hard to detect. You should be on the safe side and be immunized.
Thank you. And I'm going to ask one last question for Dr. Wilson. Since you work kind of in an international context, are you seeing a difference in how information is disseminated in the US versus how it's disseminated in other countries?
Oh, Lord, we're going to try to not be here all night, but yes, the short answer is yes. It was interesting. So of course, early on, we were looking at the countries that did exceptionally well in containing the virus. So these are countries like South Korea did exceptionally well. Even the countries that are now entering another lockdown, when you look at the European countries like France and such, what you saw there was a very apolitical message. And so when the South Korean press conferences, they started with a person at a podium, and that person was a scientist, right? Like there was no introduction, there was no context of an administration or anything. It was kind of, the response was viewed as not as something of the political government, but as something that is done for the people, in the same way that, we make roads for the population. That is not viewed as a political thing. And I think that a lot of the messaging of what needed to be done and when was stripped from the politics and was very much so presented from a scientific point of view. Not saying that that's necessarily the best way, that you need a scientist there, but the person who is presenting the information was not a representative of a political affiliation. And I think that, and the seriousness of it and the non-option of opting in was very, very clear. South Korea, they were tracking people's phones, for doing their contact tracing. Now, I'm not saying that that's what we should do here in the US. That's privacy issues, but there are other countries. I mean, you see the clear line at the Canadian border. They are doing something different. They are very closely monitoring the situation. As soon as there are a number of cases, Toronto just shut back down a couple of weeks ago. And that has been the hardest part, I would say, being here in the US. And I have quite a bit of family who live overseas. And the message was like, if we're in Africa and we can wear masks, why can't you? And like, it just wasn't even a question. And I think that the things like if you were traveling someplace, you had to have a piece of paper saying where you were going, and anyone on the street could stop you and your piece of paper had to match up with where you said you were going. And again, these countries are now very much so entering into what is their second wave, and it remains to be seen whether that ends up being, worse or better than the US at this point. But the seriousness of the general public, the unity of the public in response to trying to get this under control quickly was very, very clear in Europe, in Asia, clearly. And I would say the country that was most similar to us in policy actually ended up being the UK. And I would say their patterns of infectivity followed as such. Yeah.
Thank you. We have another question in the chat. I think this is for you, Dr. Jeff. How do you self-treat at home? I have heard about 2 medications, but they are provided to very sick patients.
Yeah, so one of the things I will say, going back to that only in many a few, you are well connected with a bunch of Black women physicians. And so I'm well connected with a bunch of them. I got the virus very early on. And so I did get a prescription for a Z-Pak, which is actually an antibacterial. But it did clear my symptoms up. And then I did a lot of, I took cold medication. So I had a very minor and mild case. So I had azithromycin, the Z-Pak, as well as some cold medication. I believe it was just Dayquil and NyQuil. And then before actually getting the virus and after, I was taking zinc, L-lysine, and echinacea. I was dropping, so those are kind of like some home, organic natural remedies to keep my immune system high. But yeah, I think I'm a person who has a very suppressed immune system when I'm stressed out. I also got shingles at 26, so I was very, very, very fortunate. But I would imagine a lot of that has changed now. And again, I think a lot of the physicians who are working with patients who are self-treating at home have much better, regimens. When I was sick, they just kept telling me to take Tylenol. Unfortunately, that was the only thing that they said. And all the other things that I told you about were things I took on my own will. So.
Thank you. So we unfortunately are out of time. I am going to ask our panelists, we did have one last question, and if you have any thoughts that you could put in the chat regarding entities you all think are doing unique segmented messaging that is penetrating the Black community, if you have any thoughts for our participants that you can put in the chat. But I want to thank our panelists again for giving of their time. They are all very, very busy women who are actively engaged in this work. And so for you all to give your time makes us feel very special. Spelman loves you, and you obviously show your love for Spelman. So thank you all again for participating in this panel, and we will see you next year when we come back in the spring with 2 more sessions. Thank you, everybody.